In this video, we'll have a look at capillary exchange. Now, of course, capillary exchange is extremely important, and this is a way that we get materials from the blood into the tissue and waste and materials from the tissue into the blood. It's a two-way exchange. Now, it's really only through capillary walls that exchanges are made between blood and surrounding tissues. That really is where the vast majority of this occurs. And once again, when I'm talking about capillary exchange, I am talking about a two-directional movement across the capillary walls or capillary walls. And of course, what type of things are moving? Well, not surprisingly, water, oxygen, carbon dioxide, glucose, amino acids, lipids, minerals, antibodies, hormones, wastes, and ammonia. So there's quite a few things that can move back and forth. So it's a very dynamic area. And when we're looking at things that move across, we can kind of break it into categories and look at different mechanisms. That if we talk about chemicals that pass through the capillary wall, well, they can, think about what they can do is they can endothelial cell cytoplasm, they can move through that and exit. They can use intercellular clefts between the endothelial cells, or they can use filtration pores, which is a concept of the fenestrated capillaries that have been mentioned in a previous video. And there's different mechanisms that actually help it to do this. And certainly we could have diffusion or the process of transcytosis, where something basically enters the cell on one side, crosses the cell, and exits on the other. So we're looking at process of endocytosis and exocytosis that's combined as things move across. And also filtration and reabsorption. So diffusion is an important component of capillary exchange, and things like glucose and oxygen diffuse out of the blood, and of course carbon dioxide and other wastes will diffuse into the blood. And capillary diffusion can occur in two different situations. One is where the endothelial cell is permeable, or this endothelial membrane, I should say more specifically, permeable to the solute. And if this is the case, then it can move through. And a solute can find passages that are large enough for it to pass through. So in some cases, it doesn't have to actually go through a cell membrane. It could use things like filtration pores, intercellular clefts, in order to move back and forth. Now, lipid-soluble substances, of course, lipid-soluble plasma membranes are made out of lipids, so they can easily diffuse through there. And some examples are the steroid hormones, oxygen, and CO2. Now, if you're a water-soluble substance, that means you basically have to use a filtration pore or intercellular clefts. And some examples of those would be glucose and the electrolytes. Now, if you're a large thing like a protein, unless under very special circumstances or very special transport mechanisms, you routinely do not leave. You're held back because you can't get your way through the cell membrane and you can't fit through any of the pores. Now, talk about transcytosis. And this requires vesicles, so it's a vesicle-mediated transport that moves materials across the endothelium. And really what happens is things get picked up by either pinocytosis or receptor-mediated endocytosis. And then what happens is, so you get into the cell, and which makes up the wall of the capillary, and now what you'll do is you'll cross it and you'll exit on the other side. So transport vesicles move across, and the materials discharge on the other side by exocytosis. Now this isn't the big way things get across. This really is the small, sort of a smaller way it gets across, but it's very vital for materials that cannot use the other processes. So if you're a fatty acid or albumin, which is a protein, or some of the hormones such as insulin, this is how you're going to get across, because the other mechanisms aren't available to you because of your chemistry, etc. Here we can just see sort of an illustration of some of the processes. Things can use filtration pores to get out. You can, if you're lucky enough to diffuse through a cell, then you can just straight do that. Intercellular clefts, okay, versus the filtration pores, or the case of transcytosis, where something will be taken in, will travel in a vesicle, and will be discharged on the other side. And of course, here's a red blood cell. As you can see, red blood cells really cannot use any of these mechanisms. And proteins such as albumin, they're too large and don't have the right chemistry to use these mechanisms easily. So when we're talking about filtration and reabsorption, this is about things such as blood hydrostatic pressure and colloid osmotic pressure. What I've done is I put together a standalone video looking at forces such as BHP, which is blood hydrostatic pressure, and COP, which is colloid osmotic pressure. So I'm going to end this video here, and feel free to go and check out that other video on the actual forces that are involved in capillary filtration.